Steve Hollenberg from Camden. He's going to speak about the physiology of the microcirculation. Every talk is going to be for 15 minutes. We are going to get a couple of questions after every talk whenever we have questions. So go on, Steve.
Thank you. It's a pleasure to be here and to talk about such a fascinating topic. So I'm charged to talk about the physiology of the microcirculation, and then we'll go on from there. I have no relationships to disclose with respect to this talk, and that is slightly different from saying that I have no relationships whatsoever. So where are we talking about the microcirculation? Here is the microcirculation is everywhere in the body, but here is the microcirculation. It is that part that goes after the large arteries, the arterioles, the capillaries, the venules, and so it excludes the large arteries and the large veins, and everything in between is the microcirculation. And the microcirculation is very important. It has a number of important regulatory functions. It regulates blood pressure. I'm going to show you a graphic about this, but the largest pressure drop in the circulation occurs at the arteriolar level. That's as it should be, and that has important consequences for regulation. The microcirculation regulates local perfusion. It puts it in one place and not another on both the, if you will, the organ level and within organs and even on the cellular level. And it does that through a couple of mechanisms that were built in, the myogenic response and also release of vasoactive substance, as well as certain other things like neurocontrol and extrinsic compression. And endothelium is very important, and most of the endothelial function is microvascular. The microcirculation is the principal site of inflammatory cell recruitment in postcapillary venules. Vascular permeability is a phenomenon of capillaries and venules, and the coagulation system is regulated by endothelium in the microvascular, and also to some extent in at least larger vessels like medium-sized coronary arteries, really a story for another day. So part of this is related to structural aspects, and what you see on this slide are the arteries getting larger to smaller as they come out of the heart, elastic arteries, muscular arteries, arterioles, capillaries, then going back into venules, medium-sized veins, and large-sized veins. So microcirculation, strictly speaking, is in here. Well, I'd like to make a couple of points with these structural aspects. The musculature, they all have endothelium, but there's musculature in the elastic arteries, the muscular arteries, and the arterioles. After you get so the arterioles have musculature. After you get there, the capillaries and the small venules don't have musculature. That implies that they don't regulate their tone. And then the medium-sized veins and large veins do have muscular. And then the endothelium are somewhat different in terms of capillaries and pores and stuff. We'll talk a little bit about that in just a little bit. So the importance of the musculature and the arterioles is shown in this graphic talking about the circulation, a schematic representation of the left heart through the aortic valve, through the aorta with an elastic artery, that's a large vessel phenomenon, and then into arterioles with variable radius. And by impacting the tone of the arterioles, you can impact the distribution of blood into different capillary beds. At the capillaries, there's exchange of various material with the cells, and then the venules, then they collect into venules, back into veins, back into the right heart, pumped into the lungs, and drained back into the pulmonary veins. So this arteriolar regulation means that there are pressure changes in the cardiovascular system. And so what you see is that there's a pressure drop. Now, the pressure has to drop from the start of the circulation to the end, right? That's necessary, at least in the mean level, to keep the blood going forward. Well, what you see is there's not a lot of pressure drop in the aorta, and there's actually not a lot of pressure lost in the veins. The predominant pressure drop over here comes in the arterioles, and then to some extent later on in the smaller arteries and the arterioles, and maybe a little bit in the capillaries. So the pressure drop is at the arteriolar level. And this slide shows the relationship between pressure and also velocity. So pressure is here in the green. Notice that it's pulsatile here in the large arteries, but sort of smooths out. It's kind of what you'd like to do. You'd like the cell to go smoothly through the capillaries so it can exchange nutrients throughout. But it also, that velocity is important. So velocity is equivalent to flow divided by area, and the flow, by definition, at all points in this system is the same. It comes out of the heart, it comes back to the right heart. The flow has to be the same, and therefore velocity is proportional to area. So the cross-sectional area of the vasculature is predominantly in the microcirculation at the arteriolar and capillary and venular level, and that means that the velocity is slowest in the microcirculation, fast here in the aorta and fast here in the veins. And if you think about it, that's actually what you want your circulation to do. You don't want it to waste a lot of time coming out in the aorta. It's got to get distributed to the organs. So you want it to go fast. Then you want it to hang around for a while and deliver nutrients to the tissues on a slow basis. You want the cells to go slowly through the capillaries. And then when they've done their job, you want them to get back to the heart quickly and be replenished with blood and nutrients. So the structural and regulatory capacities of microcirculation regulate that blood flow. So this is another graph, and on the left you see the circulation in it, and it shows that the different arteriolar beds are perfused by the aorta, and then they have microcirculation all within them to regulate. And within the circulation there are smooth muscle cells, and there are precapillary sphincters here. And then there are meta-arterioles that essentially provide a thoroughfare for blood to sort of, it doesn't have to go through the capillaries. There is instruction it can get back to the small venules and bypass this section. And there are actually occasionally arteriovenous anastomosis and precapillary sphincters. This structural feature is going to play into microvascular shunting, something I suspect you might hear about a little more before we end the afternoon. So I'm not trying to jump the gun on Jean, but sepsis is a microvascular disease. So I won't talk about the mechanisms here, but it's a disease of vascular tone, and thus is a disease of the arterioles. It's a disease of coagulation and vasodilation at the endothelial level. It's a disease of increased permeability at the capillary level, and it's a disease of neutrophil aggregation and inflammation at the venular level. So let's talk a little bit more about microcirculatory flow regulation. So here's another schematic. We've got our aorta here and our capillaries here, and we're regulating flow at the arteriolar level. And up here you have neural factors, physical factors, and then you have mechanisms of vasoreactivity control. So the neural factors are basically at the level of the prearterioles, and they have constricting influences of the sympathetic nervous system and, to some extent, dilating influences of the sympathetic system. You have physical factors, probably a little more prominent in places like the heart. Heart does have a microcirculation, important for perfusion and no reflow and all that. And there's extravascular compression of the microcirculation during systole, particularly in the left ventricle, to some extent in the right ventricle. Now when we go to reactivity of mechanisms of vasoreactivity and regulation of flow in the circulations, we have endothelial regulation, and on the left side we have endothelial regulatory dilators, vasodilators, nitric oxide, EDHF, endothelium-derived hyperpolarizing factor, and prostacyclin, and they also have endothelium vasoreactivity vasoconstrictors, endothelin, angiotensin, thromboxane, and prostaglandin H2. So that in response to shear stress, the endothelium regulates blood flow so as to achieve the most efficient distribution of blood flow to the most perfused. Then you have a myogenic response, and that's a response mediated by protein kinase, phospholipase C and protein kinase C and KATP channels, in which the arterioles regulate their tone in response to intramural pressure, again to maintain flow in response to changes in blood pressure. And then you have metabolism. This is the cardiac system, but it's also true in the other areas. Adenosine is produced in areas in which there is decreased blood flow, and that causes vasodilation. And you have nitric oxide, and to some extent, actually nitrate and nitrite as a mechanism to regulate blood flow in the microcirculation. So the other thing to talk about is blood flow heterogeneity. And this is it's a lovely paper by I believe it's a Danish group. And there's a little bit of physics there. I had to review it. It took a long time. In any case, it's a really lovely paper talking about the effects of capillary transit time heterogeneity on oxygen extraction. And the short version here is if you have homogeneous flow on the left, you have essentially equivalent oxygen extraction in all of the different capillary beds. But if you have heterogeneous flow over here, you have differential oxygen extraction, and thus the potential for different levels of efficiency and different levels at which oxygen extraction is incomplete and cellular perfusion is inadequate based on heterogeneity, so that for the whole circulation, you may have only a slight abnormality, but in different sorts of in different beds, you may have more profound abnormalities. I'm sure we'll hear a little bit about how to get heterogeneity of blood flow in the microcirculation as well. And in addition, the endothelium regulates coagulation. So here's just a schematic of what happens normally. The endothelium is designed to keep the blood cells from sticking to the walls and to allow them to flow smoothly through. And so the platelets are inhibited by various things. Clots are inhibited by plasminogen and plasmilin, and they inhibit thrombin, and that allows for laminar flow through the circulation. Again, the most energetically efficient way to perfuse a circulation. In certain diseases, you can have prothrombotic states, perhaps driven by inflammatory cytokines that cause release of fibrinogen and decreases in plasminogen and plasmin, and increases in thrombin, and you get platelets aggregating in a fibrin platelet mesh and blood clots where they don't belong. That occurs in the in the small arteries and the coronary arteries, but it also occurs in the microcirculation, and again can be an important abnormality in some diseases. And finally, you have not finally, but you have capillary exchange. So again, the blood cells are in the capillary. You have diffusion of molecules through the interstitial fluid in and out into the tissues and cells. And if you take this small section of the capillary and you blow it up over here on the right, you see there are a number of mechanisms of diffusion. You can have direct diffusion through the cells. You can have diffusion through what's called intracellular clefts. And you can have diffusion through endothelial fenestration pores, that is, pores in between the endothelial cells themselves. Normally, that is regulated. In disease states, that may not be entirely normal. And then on a macro level, this is expressed. There are forces that regulate this, and this is a graphical description of Starling forces on the capillary level. So you have blood pressure. In the vessel pushing blood out, and the interstitial pressure pushing back, so that you either have a net pressure out or a net pressure in. And then you have osmotic pressure in the blood vessel, keeping the fluid in, and osmotic pressure in the cells, sucking the fluid out. And the balance between the pressure and the osmotic pressure in both the circulation and the interstitium determines the net flow of solutes and the leakage of at the capillary level. And finally, we have neutrophil aggregation. This is another slide of that. The leukocytes have these integrins, and when these integrins are activated, these integrins interact with selectins on the cell surface, a little bit in capillaries, but really mostly in postcapillary venules. And the expression of those selectins can be induced by activated macrophages releasing cytokines and chemokines in response to microbes. The integrins eventually reach a high affinity state, and they bind. First they roll along the endothelium, and then they're activated by chemokines. Cells may have stable adhesion, and they may transmigrate through the endothelium so as to be activated at sites of inflammation. This is a regulatory process by which the presence of microbes in the circulation recruits inflammatory cells to fight infection. This is a normal process, but again, a normal process regulated in a complex fashion. You can imagine there are states where that might be, and that might be abnormally regulated. And finally, a very interesting structure called the glycocalyx. So here's the endothelial cell, and the endothelial cell is, on top of the endothelial cell is a proteoglycan matrix that sits in between the endothelial cells and the surface of fluid, and that is in some sense, it can be an important regulator, and we may hear a little bit about glycocalyx. Here it is in electron microscope in fine detail, looking at this glycocalyx sticking out of the endothelial cell, and you can imagine that plays a role in rolling of inflammatory cells and diffusion of substrates through there. So the microcirculation regulates local perfusion through the myogenic response, through release of vasoactive substances, and to some extent, at least it regulates through the microcirculation and vasoactive substances, but there are external influences like neural control and to some extent outside compressive forces. It regulates the distribution of local perfusion so as to distribute it to places of need and hopefully do that in an efficient fashion. It maintains an antithrombotic barrier, and it's the site of diffusion of molecules and fluid, and it recruits inflammatory cells. So this is the physiology of the microcirculation. You can see that that's really important. So this is the end of the talk on microcirculation, and the rest of the session is devoted to the fact that sometimes things go wrong. Thank you very much.